Okay, thank you very much for the second opportunity to present our work on the function of ceramide transfer protein in the biogenesis and the sphingolipid composition of extracellular vesicle. So the outline of the presentation, I will just give a brief introduction on how extracellular vesicles are formed. Then I will move to the hypothesis, results. It's going to be mainly about results, and then the conclusions. So extracellular vesicles are formed in the cells in the multivesicular endosome. In the multivesicular endosome or body by inward membrane budding. And this inward membrane budding can be triggered via three ways. As you see here in box one way it's through a multi-complex platform, which is called ESCRT, and it's composed by 4 subunits, 0, 1, 2, and 3. And this multi-platform complex is able to trigger the inward membrane budding. Furthermore, it's also important for the cargo selection that is going to end up in the extracellular vesicles. For instance, the ESCRT-1 subunit contains TSG101, which is going to come later in the talk. And it's able to bind to ubiquitinated protein that will be then transferred into extracellular vesicles. It can also interact with the ESCRT-2, and it's crucial for the membrane budding and for closing the neck of the vesicle. A second way in which EVs are formed is by utilizing tetraspanins, which are transmembrane protein, and it's thought that they form an enriched tetraspanin-enriched domain, which is able to trigger again extracellular vesicle formation. An example of tetraspanin are CD81, cluster of differentiation 81, or CD9. The third way is the one that actually I'm going to talk about in this talk, is the lipid way, is the lipid pathway. And among this lipid pathway, we have also the ceramide pathway, which is of interest in our lab because we know, by now we proved that there is a correlation, an association between the ceramide pathway and disease. So what we know about the ceramide pathway? Well, what is thought is that ceramide, because of its particular chemical structure, conic structure, it's able to spontaneously trigger membrane budding. And we know that there are at least 3 enzymes that are controlling this process. So what you see here, it's a graph showing EV number in percentage of control. And what you see is that sphingomyelinase sphingomyelinase 2, but not 1, it's reducing. So if you downregulate sphingomyelinase 2, you have a reduction in EV secretion. But sphingomyelinase 2 is not the only one responsible. There is also the sphingomyelin synthase 1 and 2, which if they are downregulated, on the contrary, they elicit extracellular vesicle formation and release. So this means that there is ceramide has to reach the trans-Golgi to be substrate for the sphingomyelinase 1 and 2. And sphingomyelinase has to enter the endocytic pathway to reach the membrane for being substrate for the sphingomyelinase 2. But this process, of course, points to a central role of CERT, because CERT mediates the transfer of ceramide between ER and Golgi. So it's because of CERT that actually ceramide can reach the transgolgi and potentially be involved in EVs formation. So our hypothesis was that actually CERT could play a central role in EVs formation. So first, we explored if CERT could be found in association of multivesicular endosome, MVE, which here are stained in, so this is a confocal image, and MVE was detected with LBPA, which is a marker for MVE. And in red, you have CERT staining, immunolabeled CERT staining. And what you see is that we found a colocalization. And actually, it seemed that CERT to be not only colocalizing with the MVE, but be inside MVE, as is also shown in B in the line scan analysis, where CERT is peaking right where the MVE signal coming from the LBPA it's found. So then we searched if actually CERT could be found in association of EVs extracellularly. So we isolated extracellular vesicle for the supernatant of N2A cells, neuroblastoma 2A cells, and we found that CERT was present by sucrose gradient. The isolation was performed by sucrose gradient, and we found that the CERT was found in the Alex enriched fraction, as you see here in C. So then the question was, why would CERT be there? And if CERT in EVs is actually has a function there. So what we did is to screen for possible interactors of CERT among the protein that we know are involved in the EVs formation. And we ran the two hybrid system with the pH, middle region, or the start domain as separate bait. And we found that the start domain was interact with the GSG 101, that, as you remember, is part of the ESCRT system. And we confirmed this finding by PLA assay, by proximity ligation assay, which is a technique that generates a fluorescent signal if two antigen are in close proximity. So what you have in B, it's a confocal image showing the PLA in green, so positive PLA in green, and CD19 tomato positive intracellular vesicle, meaning that the complex seems to be located in MBE. And since we found that the interaction of CERT was through the start domain, which binds ceramide, we thought to test if ceramide is controlling, it's regulating this interaction. And to do that, we depleted cells of ceramide by synthesis by using FP101, and we counted the PLA positive cell, the PLA positive per cell, and we found that they were reduced. So this means that CERT forms a complex with GSG101, potentially in the MVE, and this is regulated by ceramide. Next, we wanted to see if this complex actually is inducing, it's functional, it's inducing EVs formation. And so we ectopically express CERT, we overexpress CERT in N2A cells. And we measured the number of EVs in the supernatant. And as you see in A, ectopic expression of CERT increased the number of extracellular vesicle in the supernatant per cell. And next, we try to understand if this process was nucleus sphingomyelin 2 dependent or not. So we use GW4869, which inhibits nucleus sphingomyelin 2. And as you see here in B, the open squared and the gray squared box whiskers, GW was not able to completely abolish the CERT-mediated overexpression increase of EVs, meaning that the two pathways are not completely overlapping. And then we checked if by ectopic expression of CERT we could find more complex formed. And actually, this is what we found. So in C, you see pictures showing, again, PLA for TSG101 and CERT. And as you see, when you overexpress CERT, the number of PLA, positive PLA are increased, significantly increased. So this means that the increase that mediated by CERT of EVs cannot be completely explained by sphingomyelin production and the activity of neutrophenogomyelinase 2, but probably also the complex formed by CERT and TSG101, it's inducing EV secretion. Next, we wanted to understand if CERT engagement to the trans-Golgi was necessary for EV's formation. So if there is a kind of a vectoriality to the process. So ER trans-Golgi, as you see here, ER trans-Golgi endosome EVs. So to block this this pathway, we used a specific inhibitor of PI4P generated in the trans-Golgi by the PI4 kinase 3 beta. So we use EN10, which is a selective inhibitor of this enzyme. And we study the ceramide distribution, ceramide body-file distribution upon inhibition of PI4P generated by the PI4 kinase 3 beta. And this redistribution from ER to the Golgi of the body-file ceramide is CERT mediated. So what you see here in the vehicle condition, it's a relocation, redistribution of the ceramide body-file to the Golgi. And if you inhibit PI4P formation, and so the anchoring of CERT to the trans-Golgi, and so the relocation of ceramide to the trans-Golgi, the signal in the Golgi goes down dose-dependently. And if you then look into, and so this is the quantification in A. And so if then you measure the number of EVs upon treatment with the inhibitor, you see that there is a reduction. So by interfering with the ceramide, with CERT engagement to the trans-Golgi, so of the movement of ceramide between ER to Golgi, you're reducing extravesicle formation. But you're also reducing CERT presence in extracellular vesicles. So this is in Imbudo blot, where the CERT was normalized to flotilin. And as you see, you have less association of CERT to extracellular vesicle. So this is vesicle preparation. But next, we thought that another transfer could actually take place. A direct transfer of ceramide between ER to endosome could take place because ER and endosome have contact site, and endosome during the process of maturation, there is from early endosome to late endosome or MVV, there is an increased activity of an enzyme, the PI4 kinase 2 alpha, which produces PI4P. So again, it's offering an anchor for CERT to directly engage the endosome. So we tested this hypothesis. And first, what we did is to see if actually CERT was found in contact site of ER and endosome. And so what you see in A, it's the ER stained by calnexin, LBPA in magenta, and CERT in red. And as you also see here from the line scan, when the vesicle LBPA positive vesicle starts, you have a peak of the ER, but also of CERT, meaning that it seems to be in the right position, in really the contact site between ER and endosome. Well, but this is, of course, not enough. And so we search for a selective inhibitor of the PI4 kinase 2 alpha, which, as I said before, is the one that mediates the PI4P formation specifically, or rather specifically in endosome. And so we found this NCO3 inhibitor. And to be sure that we were not affecting the redistribution of ceramide between ER to Golgi, so the pool of PI4P in the in the Transgolgi, we tested the drug at different concentration and CO3 at different concentration in the assay of using the body pi, the Ceramide body pi fluorescent dye redistribution to the Golgi. And so what we found is that the dose 5 micromolar or below that doesn't affect the Ceramide distribution to the Transgolgi. So this means that this dose is not affecting CERT relocation of Ceramide between ER and Golgi. But this dose, this 5 micromolar dosage, was enough to reduce EVS formation and also reduce the number of the presence of the Ceramide body pi in EVs. So this means that there is a direct transport of Ceramide between ER, which is regulated by PI4P generated by PI4K2 alpha, that is mediated by CERT. And this is also important for EVs formation, because if you block this pathway, you have less EVs. So to next, to see how the sphingolipids profile would change upon inhibition of CERT activity, we use the drug HPA12, which we presume will block, it has been reported that blocks for sure Ceramide relocation to the Transgolgi, but potentially also the relocation to the endosome. And what we found is that HPA12 reduced significantly Ceramide presence in EVs, as well as sphingomyelins, especially C16 and sphingomyelin 16, but also sphingosine and sphingonin. So to summarize a little bit what I've said so far, so there is potentially two pathways in which where CERT plays a role in EVs formation. One is depicted here in blue, where it sees CERT, it's an indirect pathway because it needs CERT to first relocate Ceramide to the Transgolgi, that then can be converted or not to sphingomyelin, entering then the endosome and potentially being the sphingomyelin or the Ceramide being substrate for the sphingomyelin 2 and the sphingomyelin the SMS2 and the neutral sphingomyelin 2. But there is also an alternative pathway, a more direct pathway, which is present at ER MVV contact site, which is regulated by PI4K2 alpha PI4P production, where it sees Ceramide relocating Ceramide directly to MVE. After this direct relocation of Ceramide, a complex is formed with TSG1, which can trigger inward budding and extracellular vesicle formation. So in conclusion, we report that CERT enters the endocyclic pathway and is released in association with EVs. A complex is formed between CERT and TSG1, TSG101, which is part of the ESCRT-1 machinery. Inhibition of Ceramide biosynthesis by FP101 reduces the complex formation between CERT and TSG101. An interaction of PI4P with the pH domain is necessary for CERT to be sorted to MBV and induce AV biogenesis. PI4P produced by PI4 kinase 2 alpha controls a direct transfer of Ceramide from ER to MVE and CERT-mediated EV generation. And inhibition of CERT with the drug HPA12 reduces the formation and concentration of Ceramide and sphingomyelin EVs. And so overall, this indicates that CERT may link the Ceramide pathway to the ESCRT pathway by acting as a switch on for the CERT TSG1 complex to be formed. So now I would like to thank you, my mentor, Dr. Era Biebrich, and my lab members and our collaborator at Maastricht University, Pilar Martinez, and our funding agency. So